First of all, very good morning to all of you. I'm really highly indebted to Dr. Saxena for calling me for this meeting. It was on a very short notice, but and it was, I think he was also not that much serious when he said, but I took it very seriously when he said that I have to come back from KGMU to this place. And it was in my mind that it is the meeting on 28th, so that's why I booked my flight on 29th. Unfortunately, the same day I have to rush also. So all of us have gone through with this problem of COVID-19, and now we are all seeing that what is happening with the long COVID and now so many other things. So directly coming to the topic is that now we have seen the SARS-CoV-2 infections. So normally this is an RNA virus, which is a new virus at that time when we have seen. Normally the RNA viruses, the time period is 3 to 4 weeks, and we never thought of that the virus will sustain such long that those many years also this can, still we will have the cases. But if the symptoms persist after 4 weeks, we say it is called long COVID, and then there are the various symptoms. I'll come into the details of all these things, that how the long COVID is actually defined. But now, even after that also, if we are getting the cases in the different organs, that is called post-COVID syndrome, and now the new word has been given as PCS. So now what we are getting is the PCS cases, and the PCS cases are very dangerous, and at least 7% of them are fatal. So although the mortality is 7%, but several cases are coming in that. So that's a very important for us, that this too, how we are going to handle. There is no official protocol given. The CDC and the WHO are working. Government of India is still not that much doing, or you can say we do not have those many definitions. It's still available that what should be classified under which particular category and how it can be done. Now, what we have done, that when in 2019, the first case came in Wuhan, we got an access because we were having an international consortium of all the virology lab. So I'm a trained biosafety person from CDC and Canadian Health Science Center, and I am one of the experts for WHO. So we got these two for the first patient from the Wuhan fish market, and what we have found that they were different from the previously known SARS-CoV infections which we have seen in 2005. And that alerted us that how to start. So we had a big discussion, and some of our students were not agreeing that why we are focusing on spike protein. And this was the first structure of the world where we have seen the spike protein in an electron microscopy, that how they spike. This was very much dangerous for us, that why there is a cluster coming on this head. And that cluster was actually the spike protein ACE binding domain, and that we have characterized. So this has opened the sequencing data. All the RT-PCR in the world has been done based on this particular work. This is the world's first work, and that has been recognized by the WHO. Now it is done in all the languages of the United Nations. It is all available on the United Nations website and WHO website. It came at that time in virus disease, world's first publication in this area from our lab from India. Then immediately we thought, if this is going to happen, then what happens to the glycosylation and the structural and antigenic variation? And this was very important for us, because the glycosylation means the attachment of the glucose moiety onto the cell membrane, and that was the way by which the virus enters. So we found there were, except a very few, which were the known sites in. So this is the SARS-CoV which we have seen in 2005, and this is the new one, 2019. At that time we were not saying it's SARS-CoV-2. At that time we were saying 2019 nCoV, novel coronavirus, and we have found there were several new sites were. So this is just like a door. So we know that if someone has to enter in this room, there is one door. So that is the glycosylation site. And now if a terrorist has to come, he will never use this door. Or even if he uses this door, he will try to wear a similar kind of iCard and everything. So he mimics that, yes, I am your own. So that's how we looked into all those. So these are the iCards, you can say. So this is the previous one and this is the new, except that these two iCards were matching, the other ones were new. And that's how we came to know, this is going to become a pandemic. So first time we said in the January 2020 itself that this is going to be pandemic. WHO has declared it as a pandemic in the March, but we have said. So this has opened the new, so we characterized that spike protein, and this was the first structure of the spike protein which came from our laboratory again, and that was at 3.5 angstrom. You can see here, and that has opened the door for the vaccine. So all the vaccines of the world were actually initially based on this work that was again done in our laboratory in KGMU Lucknow, and that was again, if you go onto the Google, the Nature has recognized and several places this has been mentioned. Then of course there was a lot of theories from where it originated and how it came from. We have done the worldwide data on the sequencing, and what we have found, this all is published, because this was a very, very important work which was creating a controversial thing at that time, that from where this came from. Still I can say there is no clear-cut answer to this, that from where this came from. But it looks like now, after looking into the long COVID and the post-COVID syndrome data, that it might be a case of bioterrorism, because normally the RNA proteins do not sustain that long. So there have been an insertion of HIV NEF protein which works as an anchor protein. So that is all molecular biology. I'm not going into the detail of that. But just very quickly telling you that how the original strain has changed into the B strain, and the B strain has changes to the B lineage, and the B lineage eventually came to the Omicron, which we are still facing with. And these were at the 484 position. The one mutation was creating that much of anchoring that still we are sustaining with the post-COVID syndrome. So just one attachment is not letting the virus getting cleared off, and it has gone to each and every organ now. So then we have done a transmission dynamics, and because the initial case of, if you know, the Omicron came in the South Africa, and from there this has gone to the European part and then America, and wherever the mutations from Omicron to Alpha, Beta, Gamma and Delta has been seen, we made at that time that Omicron is going to be one of the most dangerous one, and then we characterized. So this was the original SARS-CoV-2 which we have characterized, and then the Omicron when we have characterized. Wherever you are seeing the red, they were the changes in these two strains. And we are still facing the outcome of all those cases which we are getting. This was again the world's first characterization data of the Omicron virus done in India. And WHO has recognized. The European CDC has done one day before us, but unfortunately their prediction was wrong, and the analysis which they have done was wrong. So they have taken out that data, and now this is again available on their website also. So this is again world's first data from India. So I think India should feel proud that how much work India has done that during the COVID. Now this is currently in last 6 months, we can see here that we are facing a lot of Omicron sub-variant of concerns. In India also, if you see, we are having the JNN, XBB and several other strains. This is all MolBio which we are doing in last 1 year. So this JN 1.1 is currently, but forget all these names, they are all the variant of concerns of the Omicron virus. Now, if you see the total trend line till this last month only, you can see the worldwide the significant number of cases per million were cumulative confirmed cases have been reported. But if you see the total number of new cases per million have significantly gone down in the world, as well as the total number of deaths per million has also gone significantly down. But compared to the cumulative worldwide, around 434 million cases by 2022, and around 10% of that were in India were reported. If you see the daily new cases were around in India have significantly gone down by 2023, and now we are getting hardly very few cases. You can see by just last month 2024. Now we are hardly seeing any cases. So now Government of India is not doing routine sequencing, but we have all that facility available at KGMU where we were doing the part of the INSACOG. But the new deaths compared to the worldwide, we were significantly down. So if you see here, this is the world's data. Ours was, yes, the peaks were somewhere a little high at certain places, but worldwide if you compare, we were significantly down, almost 10 times. So coming to the main point that what is and where the multi-organ involvement has happened, we were again very first in this area also, that we started looking onto the ACE2 expression, because that was that that cluster which I showed you in the first picture in the electron microscopy. So that is that cluster, and that was binding with the ACE2. So at that time ACE2 was the only receptor known. Later on we found that there is another receptor called TMPRSS2. So these were the different cases where we have seen the most of the things. COPD, CVD cases, the ACE2 was high. Hypertension and the type 2 diabetes, it was high, but in the type 2 diabetes, in different organs like liver and others, the ACE2 expression was down, and these are the different cell types where the H2 expression has been seen. So wherever the H2 expression was seen, we were able to see the virus at that time. So then we looked into the multi-organ involvement, and this was a multi-consortia thing done from India and Europe in collaboration. And what we have found that, think of any organ, we initially thought of the lungs, but we saw everywhere, brain and different parts of this, the neuroinflammation, acute cerebrovascular diseases, acute necrotizing encephalopathy, necrotizing hemorrhagic encephalopathy, and all this was because of the cytokine storm and the expression of ACE2. So then we started looking into these cytokines, especially IL-6, IL-12 and IL-15. Several of you might have gone for IL-6 testing now also, and we are still asking the people to check these parameters in the patients, especially in the post-COVID syndrome, and that's how we have given the newer guidelines for the people who are elderly, whether they should, how they, especially the vaccine has to be given, and if the vaccine has been given, whether they should be given the booster or whether they should not be given. This is also published, so all this you can see on the net.
We have 2 minutes more.
Yeah. So these are the different cell types where you can see that these are the different organs. If you think of any organ, wherever you are seeing that the expression is more. We have done huge biopsies for the ACE2 expression in each and every organ. It was full of the ACE2 expression. You can see the SARS-CoV, which we have seen in 2005. It was not in several parts, especially like, think of hilar, the aorta, testis, and see. But the SARS-CoV-2 was almost in every. So whatever you are seeing is the number of cases and where it was positive that has been shown. And this is the systemic and interpulmonary distribution. You see the systemic distribution was significantly higher than interpulmonary ones. And these are the different organs. How many cases were positive for like 30% for neurological, hepatic, dermatological. You think of any organ, it is there. Now we have looked into all those molecular cell signaling mechanism because that's how we are developing the drugs of this. So what is this happening? It is also causing the expression of a lot of new degenerative diseases, and what we have found with the help of MRI and several others, there is the expression of beta tau, alpha amyloid and alpha synuclein in the brain. What we have found that the hospitalized patients had a little lesser risk, but the people who are the severe had the highest risk. You can see here, so this line itself is telling you where the risk is higher. So by this way you can find out that even if you survived and if you are severely ill at that time, you have the higher risk of developing. In all those patients who are severely ill, the alpha-beta aggression was found in the tau phosphorylation. We have looked into the different cells in the brain. These are all patient data coming from the patients who were admitted and coming to our hospitals. The similar thing, wherever we have seen this expression, we got the SARS-CoV-2 infected brains, and you can see that was also significantly up. And so these are the patient numbers which we have given, and because of the time I'm not going into the detail of each and everything. Even the cases of dementia, DLB, PSP, FTD, AD, everything we have seen and then correlated with that expression. Now if you see in 2023-24 we have everything, but now we are having the rise in the cases of post-COVID syndrome cases. And so now we are looking into this long-lived memory T lymphocyte response that was not there at that time. And this we are believing is one of the biggest game changer into all this kind of problems. And very recently we have seen that how all the vaccines throughout the world which are available, how they are playing and what kind of side effects they are going to give. So this is also recently published. This is one of the number. If you have any questions, please do ask us. We will be always very happy through the Government of India to answer these questions. And this is all free. We always say that if you have to stop a pandemic, not only for the disease, even for the post-COVID syndrome also, then we have to make the people aware. That is demos. Demos means public. So if the people are aware of, the panic can be stopped. This is the world's first book written by me, and it has been one of the best books in all the biggest places, including WHO, Stanford, Harvard, Cambridge. You can get it at every place. And this has been selected as one of the best books in the different categories. Some of the new publications came which were from this work. And this month only, if you open this website called HCS Pharmacology and Translational Science, this is on the cover page. So neprilysin is one of the new protein which we have discovered can clear off the amyloid beta, and which we are doing in the TBI patients who were positive at that time with the COVID. And with this, I want to thank you for your precious time. Thank you very much.
Just take a seat here. Please do.